Hi, everyone, and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk Series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the online open community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. This webinar series began last year, and we are nearing our one-year anniversary. We've held over 30 events in this series so far. If you haven't already done so, check out our archive to watch all the previous talks in the series. You can find information about each event on our plantae.org blog and subscribe to our Plantae YouTube channel to get notifications when new recordings are posted. We have more weekly events lined up through April. After that, we'll be switching gears to plan and host the annual plant biology meeting online. Next week, our speakers will be joining us from China to share information about their work on plant development. Visit plantae.org/education/Plantae Presents to register for all of the events in this series. If you need to leave this webinar early, know that a recording will be made available within a few days, except for Xing's talk. If you're here today, where his is actually not going to be recorded. Today, Itumeleng is joining us to moderate. Itumeleng has a firm background and deep understanding of Mediterranean ecosystem and ecological theory. His current work focuses on understanding which assembly processes are delimiting the plant microbiome, as well as the evolutionary history of niche shifts and stability. His work will contribute to our understanding of how plant microbiomes are assembled and maintained, and more importantly, offers a new perspective on the hologenome theory of evolution. He is passionate about science communication and contributes to online discussions regarding advances and challenges in microbiome research. Thanks so much for joining us today, Itumeleng. I'm going to hand it over to you now.
Thanks so much, Katie. I'm excited to host today's session, and I look forward to engaging with everybody. Our next speaker is Jonathan Jacobs. He's an assistant professor at the Department of Pathology at Ohio State University. And his team is interested in defining the molecular and evolutionary basis of how bacteria colonize plant tissues. And his research uses novel and high throughput technologies to determine the basis of tissue-specific behaviors in host colonization and plant pathogenic bacteria. Jonathan, I'm excited to hear your talk. Over to you.
Well, thank you for the kind introduction and the opportunity. I really appreciate ASPB inviting me to present about my team's research. I'm really grateful for this opportunity to introduce myself somewhat to the ASPB community. I'm pretty new to this organization, and I'm excited to talk to you about some of our team's work. My team is interested in how bacteria colonize plants. And we really look at the plant through the lens of a bacterium. For my talk today, I'll be primarily focusing on how bacteria colonize the vascular system. But right now, I want to highlight some work from a postdoc in the group that is interested in understanding how bacteria colonize the non-vascular mesophyll tissue. And from what you can see here, that from this confocal microscopy image is that bacteria that are in yellow are dynamically colonizing the leaf space. It's actually to them likely like a large landscape of living plant cells. And the plant cells here are highlighted from autofluorescence and chlorophyll. And you can even see different cell types. You have the parenchyma tissue, those are the globular cells that you see, and then you also have stomatoguard cells. But the bacteria are associated with all of these, and also some are freely moving individually through the tissue. So we're really enthusiastic as a team to understand how bacteria colonize plants and really looking at the plant through the lens of a bacterium. Beyond tissue specificity in the group, so trying to understand what allows for bacteria to succeed in different plant environments in these microbial spaces, we're also interested in using molecular and high throughput tools for diagnostics. So translating some of the tools we regularly use for plant disease identification. But today I'll be focusing primarily on our work about bacterial plant colonization. First, I'd like to recognize the team of awesome people that really make this work possible. It is difficult even now that some people join the lab and I can't even have a photo together, as many of you know. But I'm really grateful for this team that supports this work. So Stephen Cohen is a postdoc of the team that took the image that you saw before, but I'll be primarily talking about work from a student in the team, Jules Bouchakas, and postdoc Vero Roman Reina. And so I'll be highlighting some of their work today about tissue-specific behavior in plant pathogenic Xanthomonas species. In the team, we focus on Xanthomonas bacteria. This is a genus of bacteria that infects over 400 plant species. It's really important for agriculture, but these organisms also infect plants in natural ecosystems. And the first thing you might notice are the different plant species that they infect. It actually makes for an incredible model for understanding host preference by microbes. And what I'd rather have you focus on here, though, are the symptom types. So Xanthomonas pathogens cause vascular and non-vascular diseases of plants. Some of these are depicted here. You have vascular wilt of banana. You have black rot of cabbage. These bacteria enter through hydathodes and then colonize the xylem. I would recommend checking out Laurent Noel's work from LIPM Toulouse to learn really about the dynamic infection process of these Xanthomonas pathogens. This is a great model to understand that. Xanthomonas oryzae on rice. These organisms just represent some of the few that cause vascular infection. You have examples, though, of short lesions or spots that develop from some Xanthomonas pathogens, and that's because they colonize the non-mesophyll or the non-vascular mesophyll tissue. They remain restricted to the site of infection. So you'll get short leaf streak development on rice, or you'll see spots develop on tomato, or even cankers on citrus. So where these pathogens colonize, whether they go to the vascular system or the non-vascular tissue, can have pretty important outcomes for plant health. One of the models in our team that we study is Xanthomonas translucens. These organisms infect cereals, and here's one organism that we work with, Xanthomonas translucens, on barley. It infects seed. It also causes blight on leaves or small leaf streaks. So it actually has a pretty diverse ability to, or it has a pretty wide range ability to infect diverse tissue types. So whether it's the seed, could go to the non-vascular tissue or vascular tissue. So it provides an interesting model to understand tissue specificity because it can go to many different tissue types. Today for my presentation, when I'm describing vascular infection, your favorite organism you work with or your tissue in the plant might be the vascular tissue you might work with might be the phloem, but today I'll be focusing on organisms that go to the xylem. So this is the water transporting tissue. It provides structure to the plant. These themselves aren't living cells, but they're surrounded by living tissue. And so you can see here Xanthomonas translucens colonizing barley xylem vessels, and it appears that they're clogging them, which restricts water movement. When I talk about non-vascular infection, I'll be speaking about bacteria that colonize the mesophyll tissue. And so these intercellular spaces where you see the bacteria, these are GFP expressing bacteria surrounding plant cells. This is a photosynthetic living plant tissue, and these bacteria thrive in this environment. but it's very distinct from the xylem, which is a dead plant tissue that has limited nutrients in comparison to the mesophyll. But these organisms are highly adapted to both or either one or the other. So in my team, we are really curious about what allows Xanthomonas to go to plants, but also could we use this as a way to tease apart questions about how Xanthomonas species go to different tissue types? Could we use the wealth of genomes that have been produced by our community to kind of nail down some traits that are associated with vascular and non-vascular pathogenesis? Here's a phylogenetic tree of Xanthomonas species. And as you can see that from this phylogeny is that many different Xanthomonas species have different vascular or non-vascular symptom types they produce. So you could imagine one scenario where some vascular pathogens group together and others, or you could have two clades, one with just vascular pathogens and another with non-vascular pathogens. But the reality is that actually that behavior is distributed across the genus. And it's not even associated with a particular host. You could also imagine that only certain plants get certain symptom types. But both monocot and dicot hosts can display either symptom type depending on the strain that's infecting them. And Xanthomonas is a great model to test questions about this tissue specificity because you can have closely related strains within a same species that cause either vascular or non-vascular symptom types. This occurs in barley. You can see this in cabbage, rice. There are many examples of this in Xanthomonas. So you could imagine the spectrum of infection. There's vascular and non-vascular, or systemic and non-systemic infection, and this results from being able to colonize the vasculature or the non-vasculature. But maybe a certain organism falls on this spectrum. So could we use these genomes to pull out some traits that were associated with either behavior? So I work closely with my colleague Alvaro Perez Quintero in Jan Leach's lab at Colorado State University and asked, are there any specific traits that are associated with tissue specificity? And so we looked across many Xanthomonas species, even Xanthomonidaceae bacterium Xylella fastidiosa. Many of you may know about this organism because it's currently wiping out olive production in southern Italy. But these are slow-growing vascular pathogens. So they share a phylogenetic relationship with Xanthomonas, but also cause vascular disease on many different plant hosts. And working with Alvaro, we asked the question, well, is there an enrichment for a particular protein that's found in vascular or non-vascular bacteria? And what we found was that with Bayes traits analysis, so it lets you briefly figure out and if there's an enrichment of a certain trait, like a protein, with a particular phenotype. So in this case, it was vascular pathogenesis. And we found that actually only one was enriched for in vascular pathogens, and it's called CbsA. So CbsA encodes it is a cellobiohydrolase. It's only found in vascular Xanthomonas species, with a few exceptions of non-vascular pathogens that have it. And it cleaves off the ends of cellulose to produce cellobiose, which is two glucose monomers. And it's speculated that this is important for bacterial growth. Actually, early media to isolate Xanthomonas species included cellobiose. So what can CbsA tell us about the evolution? Can we use this protein to track the evolutionary history of vascular and non-vascular pathogenesis in Xanthomonas species? So just to summarize, we see this tight association with being vascular with having CbsA, with some exception of some non-vascular pathogens having it, but a majority have that are vascular have CbsA. And so can we use this association to to ask what I had said, to ask about the evolutionary history, like a chicken or the egg question. What came first, vascular or non-vascular pathogenesis? And I would say that it's more complicated than just one or the other. So I worked closely with a colleague down the hall and his PhD student at the time, who's now at Penn, Emil Glucktaler in the slot lab. And we were wondering, using tools that they typically use to trace evolution of biosynthetic cluster or secondary metabolism clusters in fungi, can we use that to understand the evolution of vascular pathogenesis by following CbsA? And so I'm going to walk us through the analysis that Emil had done looking across Xanthomonas species. So these are in the upper group in this phylogeny, but also in some beta proteobacteria like Ralstonia solanacearum, also also a vascular pathogen of plants. And what we see is that the evolution is dynamic. We first looked at if there was any gene loss. Could that tell us about if organisms have lost this and maybe lost the ability to cause vascular pathogenesis? And what we see is that in X here, there are many events across the history of this organism that CbsA has been lost. So it's been inactivated or completely excised from the genome. And I'll give you some specific examples of this. So when you look at some of those comparisons that I had mentioned before, as an example, Xanthomonas translucens, you see CbsA is disrupted in non-vascular Xanthomonas translucens species. So in the 5' region, there's a transposase that's inserted and only 3' fragment is left. But in vascular pathogens, you have an intact CbsA gene. The same goes for rice pathogen Xanthomonas oryzae. In Xanthomonas oryzae, Pathovar rhizicola, the non-vascular pathogen, there's a short deletion in CbsA, but also some point mutations that completely inactivate this gene, this protein that would be if it were synthesized from this gene. But the vascular pathogens have an intact copy. So there are multiple events that have happened where actually non-vascular organisms have lost this. So we're speculating that actually an ancestral behavior was vascular for Xanthomonas species. Also, when we look at horizontal transfer, there were many horizontal transfer events that have occurred. So that would suggest actually this trait for vascular pathogenesis is fluid. So there are multiple events where we see that CbsA, for example, is transferred into Xanthomonas campestris, a cabbage pathogen, or to other species. And so what we speculate here has happened is actually there's flexibility on the lifestyle that some bacteria could transition back to being vascular. What was really exciting to see is that this gene was even transferred outside of Xanthomonas into other important plant pathogens like Ralstonia solanacearum that causes bacterial wilt of many host crops, especially tomato. And it was transferred from gamma proteobacteria to this beta proteobacterium. And we know from others' work that this is an important gene for full bacterial wilt pathogenesis. So there are clear associations with being a vascular pathogen and having CbsA, losing it with being non-vascular, but also acquiring it if you are a vascular pathogen or a pathogen that is potentially now vascular. So going back to that model I showed before, we see that the spectrum of disease from vascular to non-vascular and the evolution of that is dynamic, where you have cases where you have gene loss, where strains that are now non-vascular have lost this trait, but also some cases where organisms have acquired it. So what about function? What does this do for the biology of this organism and how it causes symptoms? So there's a long history of work that's been done by my colleague Ramesh Santi, and their lab has kind of pioneered understanding the function of CbsA in the plant. So when you knock out this gene from rice pathogens, Xanthomonas oryzae, you completely disrupt the ability to cause blight pathogenesis. So there's a severe reduction in the ability to cause blight. Working with colleagues that focus on Xylella fastidiosa, when you mutate CbsA, there is you dramatically reduce vascular pathogenesis in this organism. So there's an important role for CbsA in vascular pathogenic bacteria. But what happens in our model system, Xanthomonas translucens? As I mentioned before, Xanthomonas translucens, pathovar translucens, this organism colonizes the vascular system to cause blight. We actually use a leaf clipping method to let the bacterium enter the vascular system, colonizing the xylem. These are GFP expressing bacteria that leads to blight development. What's really nice about translucens is that we also have other strains that don't go to the vascular system. They cause non-vascular symptoms, but not xylem infection. And these are in the pathovar ungulosa. These are wheat and barley pathogens. And these organisms are a great comparative model to have to compare vascular to non-vascular pathogenesis. Again, to emphasize where these microbes are localized, Xanthomonas translucens colonizes the xylem. We don't know if clogging the xylem is part of the blight development, but we're speculating that it reduces water flow. And you can see here that when we do a leaf clipping, these bacteria move in the xylem. Often you can even get non-vascular infection by this pathogen. And so this organism kind of has a dual lifestyle, but it has the ability uniquely to go to the xylem. And if you zoom in, these bacteria are within vascular elements, and that's how they move. When I talk about Xanthomonas translucens ungulosa, this pathogen, keep in mind, only goes to the mesophyll tissue. It only goes to the intercellular spaces of this living plant leaf space. And the non-vascular pathogen has lost CbsA, so it does not have a functional copy of this gene. So what happens with leaf blight development for Xanthomonas translucens versus pathovar ungulosa? When you do leaf clipping, you get long distance leaf blight development. With the ungulosa pathovar, you don't get any, maybe barely, symptoms develop. But what happens if you express CbsA from translucens in the ungulosa pathovar? Surprisingly, we were actually really excited to see this, is that CbsA allows for XTU to go to cause vascular blight development. And if you remember, this gene is found across Xanthomonas species. Could you use another copy of CbsA from another organism to see if it has functional similarity? And it also allows for this organism to cause vascular blight development, saying that there's some sort of functional conservation or conservation of function for this protein across the genus. But does CbsA allow for xylem colonization? Is it just going everywhere when you add it to the non-vascular pathogen, or is it actually staying restricted to the xylem? So I worked closely with colleagues in Toulouse. This is work from Aude Cerruti, and what we see is that vascular pathogens, Xanthomonas translucens can go to the xylem. That's part of why it can cause blight. The non-vascular ungulosa pathovar cannot go to the xylem. But when you add CbsA, it actually confers the ability to go to the xylem. So we're now working to try to figure out the function of this. Of how does this cell wall degrading enzyme promote the ability for an organism to go to the xylem? So going back to our model, we've been able to show here that addition of CbsA to a non-vascular pathogen allows for vascular colonization. And then from other colleagues, collaborating with colleagues, loss of it disrupts the ability to colonize the xylem. So what happens with translucens when you inactivate CbsA? Does XTT lose the ability to cause vascular pathogenesis? And this is work by Jules and the team in Vero. And what we were actually surprised to see is that when you knock out CbsA, you actually don't see any loss of disease development. Jules was pretty disappointed when he started working with this because he was like, oh, how does this work in our model? And what was surprising was that he actually, just instead of only looking at the blight symptoms, decided to also look downstream of the blight. What did the symptoms look like? And what he saw is that when the organism lost the cellulase, a bunch of non-vascular water-soaking spots appeared. This rarely happened with the wild type strain. And what this looks like is in the blight, in the wild type, you get this leaf desiccation. But with the CbsA mutant, you see all these water-soaking spots. So loss of CbsA actually makes this organism more non-vascular. And what we're still trying to figure out is when you lose CbsA, even though you can still go to the xylem in this organism, what allows for it to have increased ability to go to the non-vascular tissue? So hopefully I've been able to describe to you today about the spectrum of disease development, that it lies somewhere on being systemic or non-systemic, and that's tightly associated with being vascular and non-vascular. And in Xanthomonas species, although it's not probably the only thing, this gene, CbsA, has played an important role where you see natural loss of CbsA in non-vascular pathogen, some events where it's been acquired, and in some cases where loss leads to complete inability to cause vascular pathogenesis, or like we see in nature, non-vascular host colonization. So with that, I'd like to thank my team. This is our new reality. So before I showed you, we were all in person, now we're online. I like science art. So if you like this kind of stuff, this is bacteria colonizing the vasculature. I'm really grateful to our funding sources and clearly our collaborators across the world, and especially the funding that let me start thinking about this for my postdoc. I'm really grateful for that and the advisors that let me start building some of these ideas. And with that, I'll take questions.
Thank you so much, Jonathan. Those amazing pictures, by the way. Who took them? In your lab right now? Or?
Yeah, it's a mix of me, a couple of the lab members, Jules, Stephen, but also I learned a lot of this in collaboration with Laurent Noel's group in Toulouse. So yeah, we have a confocal in my lab, and we're trying to start probing a lot of questions about bacterial pathogenesis with imaging.
Oh, fantastic. I look forward to it. We have a couple of questions for you, Jonathan. One, Gita wants to know if what are the advantages of a single gene like CbsA having a major effect on tissue specificity? Are there any other enzymes that are associated with this vascular lifestyle?
So we don't know what other genes are important for being a vascular pathogen that are unique to being a vascular pathogen. This was the only one that came out. I actually, when I first started grad school, I was really curious about this question. We only had 8 pathogen genomes, and I did pairwise best blast analysis when I was learning to code. And I actually came up with the same gene when we only had 8 genomes. And even now, when we have thousands, we only come up with CbsA. Now, I will say that there's a diversity of type 3 secreted effectors that are different between vascular and non-vascular pathogens. And a clear example of that is the sugar transporter, sweet transporters that are induced by tal effectors. We actually have evidence on rice that I didn't show here, is that actually that organism, when you add CbsA alone to Xanthomonas oryzae, Pathivara rhizicola, it doesn't do anything. But if you add it in tandem with the sugar transporting inducing sweet, it allows for blight pathogenesis. So in that case, that organism actually needs multiple factors. And it's funny that it's centralized around the theme of disaccharides, though, like cellobiose or sucrose. So I hope that answers the question. I also apologize, the cat in my house, my cat wants to get in the door right now and it's meowing. So if you hear a cat meowing, that's why.
So shortly related to Ryan wants to know if you think there's a fitness cost to acquiring the CbsA gene in your bacteria?
Yes, so we're starting to look into that. So there's work from Ramesh Santi's group that shows that this enzyme itself can induce plant immunity. But now we're trying to we're trying to understand what that fitness cost is. So, and we think that it's not only just plant defenses, it also has to do with some of the bacterial behavior. So we're trying to figure that out now, but because that enzyme itself triggers plant defenses, we're speculating that maybe that's higher in the non-vascular tissue compared to the xylem, which is dead. So maybe the plant can't sense it when it's in the xylem.
We have two more questions. One is, do vascular colonize Xanthomonas, are they more pathogenic than the mesophyll colonized in general? Do you think so?
I think they're just different. So this is about host fitness. I think actually it connects to the last question even, is that if you're a vascular pathogen and you cause aggressive pathogenesis because of the tissue colonize, it kills your host because it can. Yeah. There could be a negative fitness effect for the pathogen. So that's like a bigger question, I think, to ask. But I don't know if one is better than the other, but there is variation in how aggressive they are. But I think that just depends on the lens of the organism, right? Because those non-vascular pathogens are succeeding fine.
They're doing quite well, yeah. Just sort of related to that as well, do you find any differences looking, for instance, if you compare monocots to dicots in terms of disease presence and pathogenicity?
So I think, that is a question my colleague Alvaro, who did that analysis, is looking into, and we have not explored that, but vascular and non-vascular pathogenesis happens in both monocots and dicots. So we haven't dug deeper to see if there's an enrichment of genes in a certain kind of host type.
Oh, okay. We have one last question. Is overexpression of a single gene which comes from the oh no, that's just a comment. But other than that, okay. I was wondering, when CbsA is expressed in XTU, does it still infect the mesophyll as well as the xylem, or does it strictly infect the xylem tissue?
So we haven't really dug into that, but it can cause vascular pathogenesis.
Yeah.
It causes non-vascular pathogenesis still, but we don't know if it looks different than the non-vascular pathogen. That's basically, I was confusing myself when I was trying to talk about this. So we know that it can still go to the non-vascular tissue, but we don't know if it looks different than the wild type strain.
Oh, okay. Just one last question, because for the microbiome people, do you see any differences in colonization patterns when you have a different composition of the microbiome, not just a specific strain? Because that's something that I'm quite interested in, is to see if whether other microbe interactions could be playing a role in colonization patterns.
So that's a really, I think, cool question to even ask. Like, maybe someone can correct me, but I feel like this would be an exciting model to see what, if there are associations between being a vascular pathogen and the microbiome that's present, and a non-vascular pathogen and microbiome. Clearly there will be differences, but for me with the microbiome, what I'm really curious about is, where does this gene come from, right? Other microbes in the microbiome that we haven't sampled probably have this enzyme, and are they contributing to gene flow? That is, I think, a really exciting question to ask.
Thank you so much, Jonathan. I think that's the last question that I had. If there's anything else, Katie, over to you.
Yeah, thank you, Ichimile, for being such a great moderator, and thank you to our audience for all these great questions and kind comments in the chat. It's really great to see where everybody's joining from, and thank you for all your support of organizing these webinars possible. Thank you to both of our speakers, and I guess we'll see you next time.